Dear students, now I would like to continue the description of the different basic tissue types. Today I would like to talk about the connective and supportive tissue and the muscle tissue. In the first diagram, in this first picture, you can see a schematic picture of a loose connective tissue. The loose connective tissue is one type of the connective tissue, but here we can talk about a very huge range because, for example, the blood, it is one type of connective tissue, and the bone is also one type of supportive connective tissue. So here you can see that how big difference we have between the different types, and it is based on the different compartments. We have, for example, one type of connective tissue when the cells are the dominant. We have another where the fibers are the dominant, and that's why we have a very huge, very big change and very big difference between the different connective tissue types. Very important to know the main compartments of the connective tissue. The connective tissue consists of cells, first cells, and between the cells in the intercellular substance we can talk about fibers, and we can talk about ground substance between the fibers and different adhesive molecules which will fix them together. Here in this schematic picture we can see collagen fibers, we can see reticular fibers which will form a meshwork, or elastic fibers which are thinner, and between them you can see the different cell types of the connective tissue like fibroblasts, fibrocytes, fat cells. What I will show you now, I will talk about these compartments. So let's see now the description of the different cells of the connective tissue. So first, I would like to talk about the fixed cells of the connective tissue, which means that they do not migrate. They are there, they do not change the position. Very important, the first cell of the connective tissue is called the fibroblast. The fibroblast is an active cell which will produce the fibers and the ground substance of the connective tissue. Here you can see the picture from the fibroblast. It is a cell culture with gentian and violet, but if you see with cells with hematoxylin and eosin stainings, they are also bluish because they are basophilic. Why? So you can see here the cells, these big cells with processes, they are the fibroblasts, active cells. They produce a lot of fibers, and for the production, for the protein fiber production, we need rough endoplasmic reticulum in the cytoplasm. So that's why they will be basophilic with hematoxylin and eosin staining. In this cell culture, you can see also elongated inactive cells. We call them fibrocytes. We could have a difference between them. I mean, from the fibroblast, could be the fibroblast will be inactivated, they will be the fibrocyte. But for example, if we have a wound on the surface of the skin, there the inactive fibrocytes, they are able to activate and they are able to produce new fibers to help in the healing of the skin. After, the second cell type here, fixed cell of the connective tissue, is the reticulum cell. I will talk about later the reticular connective tissue. The reticular connective tissue, it means that they form a meshwork. The reticular fibers, they form a meshwork. They help to keep together the different cell types. For example, in the lymphatic organ, the lymphocytes, or in the bone marrow, the different developing cell types. So between the cross points of these reticular fibers, we can find the reticulum cells, which will produce the fibers. After, the third group is the group of the adipocytes. The adipocytes, or fat cells, I hope you know what is the function of the fat cell. We can talk about white fat and brown fat cells. What does it mean? The white fat cells, they are those which will store the fat within one locule, which we can say unilocular fat storage we have in the cytoplasm. And they are really important to support and protect the organs. It has a really important mechanical function and protect the body against the heat loss and physical trauma. But we have to mention another adipose tissue, which is a brown fat. In the brown fat cells, the fat is stored in many smaller fat droplets. And you can see also that here, that the nuclei, they are in the periphery. And these type of fat is really important in the thermoregulation. So we have a huge amount in newborns and, for example, animals who are sleeping during the winter time. So this is another special cell of the connective tissue. And after mesenchymal cells, we can talk about the different cells, the multipotent cells, which are able to differentiate and will form the other cell types. And finally, we can talk about the melanocytes, which are also located here in the hair follicle with the special mesenchymal pluripotent cells. These melanocytes, they produce pigment, that's why we can find it in the hair follicle, but they have a different origin compared to the other connective tissue cells, but we have to mention them. We can find there in these structures this pigment-producing cells, melanocytes. After the very huge group as the group of the mobile cells of the connective tissue, they are able to migrate in the connective tissue. These are the histiocytes. They are special macrophages of the connective tissue originated from monocyte. They are able to phagocyte different foreign particles. They are really important in the protection of the body. And also we have to mention here the group of the white blood cells, the lymphocytes, B lymphocyte, T lymphocyte, and the granulocytes, for example, the neutrophil and eosinophilic granulocytes, which are also really important in the protection of the body against bacterial inflammation, virus inflammation, these are really important in the allergical reactions. But these details you can hear in another lecture about the blood, when we will talk about different cells of the blood. And finally, I would like to mention the mast cells. The mast cells, they are also located in the connective tissue. Here we have heparin and histamine in the granules, and they are really important in the allergical reactions. After, I would like to show you the fibers of the connective tissue. In the connective tissue, we can talk about 3 different fibers. The first is the collagen fiber. Here you can see the picture from collagen fiber. It is a dense collagenous connective tissue. Between them you can see the fibrocytes. These fibers, these are the most abundant protein in mammals. We have more than 20 types of collagen fibers in the body. They have a very great tensile strength, and they are inelastic, and they will form different bundles. Mostly they are straight, but they could have a little wavy shape depending on. With hematoxylin and eosin staining, they are eosinophilic. And which is really important and interesting characteristic, that we can dissolve it with a long cooking. And maybe you heard about the Hungarian jelly, which is called the kocsonya. What the Hungarian people, they do it during the winter time. They cook for a long time different skinny parts of the pig, and after they put it to the snow, and this will form like a jelly-like material. This is called the kocsonya. It is a very special Hungarian food. I recommend you to try this food. After, we can talk about the production of the collagen fibers. The collagen fibers, they are synthesized by the fibroblasts. The fibroblasts, they will produce tropocollagen molecules, which will form a very special shape. We have very special connection between these fibers to form the fibrils. And it is really important that the vitamin C is necessary for this collagen synthesis. And the disorder is called scurvy. The disorder of the lack of vitamin C is the scurvy. It was a very special disease, you know, in sailors when they had no fresh vitamin C support in a long ocean trips. After the elastic fibers, they can stretch and snap back. It is a very special characteristic, and they can go back to the original length, and they can form different ligaments, they can form different membranes, and we have a huge amount of elastic fibers, for example, in the skin and also in the lung, when we need, you know, dilatation and very big change in the volume. It consists of a homogeneous part which is called elastin, and we have fibrillin around. And in this picture, you can see this is from the wall of the aorta, which consists of a lot of smooth muscle cells. You will learn it. The smooth muscle cells are able to produce elastic fibers, and they will form elastic membranes. And this picture is brown because we use a special staining for the elastic fibers, which is called the orthane. The other fiber is the reticular fiber. I told you that the reticular fiber consists of type 3 collagen fibers. I told you that we have a lot of different types of collagen fibers. The type 1 collagen is that what we can find in the tendons, in the ligaments, in the bones, what I told you. The type 3 is that which will form a meshwork, and that is, we say, reticular fiber. It consists of a lot of carbohydrates. That's why the special staining of it, a PAS, I will show you that slide later in the reticular connective tissue. But this slide is from the liver with silver impregnation, because the other special staining of the reticular fiber is the silver impregnation. You can see these dark structures. These are the reticular fibers in the liver. We can find a lot of reticular fibers in the parenchymal organs to keep together the cells. Okay, and after the third main compartment is the amorphous ground substance, what we can find between the cells and the fibers. This is like a jelly where we have fibers and where we have cells which are fixed to this amorphous ground substance. It has really important function during the different special mechanisms, special change, you know, in the materials, they can pass through easily, and this very special background will fix into this amorphous background will be fixed the fibers and also the cells too, with special adhesive molecules, what you can see here. But this jelly-like material consists of special molecules which are called the proteoglycans. The proteoglycans, they have a core protein and very long glycosaminoglycan chains. They are attached there to there. And here you can see one molecule, and a lot of proteoglycan molecules, they will attach to another molecule which is called the hyaluronic acid. So this very special three-dimensional molecular meshwork will form this amorphous ground substance, the basic of the connective tissue. And of course, it could be different depending on the connective tissue exactly. We could have different disaccharide units depending on where we are. Okay. After, I would like to show you some examples of the different types of the connective tissue. First of all, we can talk about embryonic connective tissue, what we can find in the embryo or newborns. Like the mesenchyma is a very undifferentiated connective tissue types during the development of the embryo. But the other I showed you earlier, this slide from the umbilical cord, this is called the Wharton's jelly or embryonic connective tissue, which consists of course, fibrous cells, glycosaminoglycans, so amorphous ground substance. But within the amorphous ground substance, we have a lot of water. And this jelly will surround the vessels, what we have here in the umbilical cord. And the function of this jelly is to protect these vessels against the mechanical effects within the uterus, within the cavity of the uterus during the pregnancy. After, if we see the different connective tissue types, we can talk about connective tissues where the cells will be the dominant. Cell-rich connective tissue is, for example, the reticular connective tissue, what I mentioned, where we have reticular fibers. Between the reticular fibers, we have the reticulum cells. And these reticulum cells will produce the ground substance and the fibers itself. And this is a special in the bone marrow, spleen, lymph node. After, a special spinocellular cell-rich connective tissue we can find in the ovary and the uterus. We will learn this in the second semester. And the areolar connective tissue, which is located between the two layers of the mesentery, where we have a lot of fat cells, connective tissue cells. This is a very special fat staining, where you can see big round shape red cells. These are the fat cells between the two layers of the mesentery in the areolar connective tissue. The other huge group of the connective tissues, the fiber-rich connective tissues, which means where the fibers are the dominant. I told you that we have a very huge range between the loose connective tissue and the dense regular connective tissue. The loose connective tissue, what we can find everywhere in the body. I wrote here to fill the free spaces. This is, for example, the wall of the intestine there. It will surround the vessels, the nerves. So it's a very huge, very big amount of tissue in the body. And the dense regular tissue is, for example, what we have in the tendon. So both of them, they are connective tissues. And you see how big difference between the loose and dense connective tissue. And between them, of course, we have a lot of different other connective tissues, depending which organ we are, what's the function of this connective tissue exactly. The dense regular connective tissue is visible in this picture. I told you, for example, in the ligaments, in the tendons. We can talk about regular dense connective tissue. In here, it's a regular one, but we can talk about irregular form of it. For example, in the skin, where the collagen fibers, they will form like a meshwork. We can see, if you see your skin, there are small lines to show the direction of the collagen fibers. And it is really important that the surgeons, they have to follow these lines, because they can help. If we put, for example, if we cut parallelly with these lines, they can help in the good healing of the skin after the injury. After, we have another type of connective tissue when the amorphous ground substance will be the dominant. And this type of connective tissue, we say supportive connective tissue, because they are stronger if you see the mechanical characteristic of them. So one of these tissues is the cartilage. Here you can see a picture, hematoxylinosin stain slide from one of the cartilage, which is the hyaline cartilage. Here also we have cells, we have intercellular substance with fibers and ground substance. The cells, you can see they are round-shaped cells with pale cytoplasm because they store a lot of nutrition inside, carbohydrate and fat, which are not stained with hematoxylinosin staining. They form smaller groups which are called chondron. And these cells, they will produce the fibers and the ground substance of the cartilage, which is called the territorial matrix, which is located around. And here is the intercellular substance, which is basophilic in this case, of course, because of the lot of proteoglycans. They have negative charge, these disaccharide units, and that's why they will contain a lot of water, and of course they will be basophilic with staining. What type of cartilage we have? We can talk about hyaline cartilage, fibrous cartilage, and elastic cartilage. Let's see these different cartilages. The first, the hyaline cartilage. Where do you have hyaline cartilages in the body? On the articular surfaces, in the trachea, in the nose, main parts of the larynx. And it is really important, the endochondral bone formation, what we will see later, it is also originated from hyaline cartilage. Here you can see the picture with hematoxylin, eosin staining, and in this part we can see a pus stained hyaline cartilage. I told you that the pus stains the carbohydrates, so it is really visible that also the intercellular substance and the cells itself, they are also pus positive. Why? Because of the carbohydrates. The cells, they store a lot of nutrition. And we have a lot of proteoglycans, glycosaminoglycan chains, disaccharide units within the intercellular substance. That's why it is pus positive. But why is it important that these cells, they store a lot of nutrition? Because here in the cartilage we have no own blood vessels. The cartilage is supplied by diffusion through the perichondrium, which is located around the cartilage. The perichondrium will help in to supply this tissue by diffusion, and it is also really important in the growing of the cartilage. I told you the cells, the chondrocytes with chondron. Here in the hyaline cartilage was the origin of the name hyaline because it's like a glassy. It is transparent. We have type 2 collagen fibers in the intercellular substance because these collagen fibers, they have a similar refractive index like the background amorphous ground substance. And as I told you, here we have a lot of glycosaminoglycans, and that's why it is pus positive and basophilic with hematoxylin eosin staining. Let's see the other cartilages. The next group is the fibrous cartilage. The fibrous cartilage, in the fibrous cartilage, we can find type 1 collagen fibers between the cells, and that's why it is much more stronger. So we can find fibrous cartilage where we have bigger mechanical effects. For example, in different articles, like in the shoulder girdle, the discus manis within the joints, and the intervertebral disc within the vertebral column. There we have fibrous cartilage, and you can see that the cells, they are located within a row, within a chain, and the collagen fibers and the amorphous ground substance is located between them. We have no perichondrium in this type of cartilage. The third one is the elastic cartilage. Elastic cartilage we can find in the external ear and also the epiglottis. This is one of the cartilage of the larynx. And here you see also perichondrium, but between the cells in the intercellular substance, the elastic fibers are the dominant. And here it is really visible with the special staining, with the orthine staining, which will show you the elastic fibers between the cells. So these were the cartilages, and let's see now the other supportive tissue, which is the bone. Here in the bone we have, which is special, that we have inorganic amorphous ground substance, which is special, and that's why it's much more stronger. It will form the skeleton of the whole body. What are the cells, what we can find here in the bone? We can talk about osteoprogenitor cells. Osteoprogenitor cells means that from this can differentiate all of the other cell types of the bone, can turn into osteoblast, and they can originate from the inner layer of the perichondrium. The osteoblasts, they will secrete the fibers of collagen fibers, which will form the main part of the bone, and of course the amorphous ground substance of the bone, which is called the osteoid. Here you can see this eosinophilic part in the slide. This is the osteoid, the organic amorphous ground substance of the bone with collagen fibers. And you see that on the top of this osteoid, on the surface here, we can see the basophilic cells. These are the osteoblasts, and these osteoblasts, they will produce the osteoid, they will form the bony tissue. And of course, when they will produce more and more osteoid, after they will be embedded within the osteoid and will be inactive. And in this case, we can talk about osteocytes, which are inactive. They are located here within this small space, which is surrounded by osteoid. It's called the lacuna, lacunes. And they have a lot of processes which will form connection between each other, because it's really important that they will communicate with each other with small canaliculi. And the connection here, you can see these cells, these inactive cells, they are really important in the regulation of the blood calcium level. And the osteoclasts, they are also really important cells of the bone, because they will destroy this osteoid, and they originate from monocyte, and they are also really important in the regulation of the blood calcium level, because they destroy bony tissue, and that's why, because they destroy them, the calcium will be free and will increase the blood calcium level. There are a lot of hormones which will also regulate this calcium level. You will learn it in physio. And the intercellular matrix, of the connective tissue consists of osteocollagen, type 1 collagen fibers, and amorphous ground substance. I told you that we have organic part, and also we have inorganic hydroxyapatite crystals which will form it. And the adhesive molecules, the glycoproteins, the osteocalcin and osteopontin molecules, they are also really important in this tissue. So let's see now the structure of the compact bone. The compact bone, this is a major bony tissue. You can see the units of the compact bone is called the osteon, which is a small unit, round shape unit like a column. In the middle we can see the Haversian canal with the vessels and the nerves. Around that, in the circular shape, here we can find the osteocytes. You see the projections which will communicate with each other with the canaliculi, and between them we have the intercellular substance with the collagen fibers and the organic and inorganic part of the bony tissue. These laminae, they are called lamina specialis around, and because of this very special position of the intercellular substance, that's why the bone will be very strong and can form the whole basis of the body. And of course, between the Haversian canal, we have communication. What we can say, these are the Folkman canal. After, I would like to tell you some words about the bone formation. We can talk about primary and we can talk about secondary bone formation. The primary, it means that the progenitor cells originated immediately will come from the blood. The mesenchymal cells immediately will start the ossification. Now we have to concentrating on the secondary bone formation, which means that we have a background tissue. It could be connective tissue or hyaline cartilage. And if the whole ossification will start from the connective tissue, this is called the intramembranous ossification. And when we have hyaline cartilage, like the background, this is called the endochondral ossification. Where do you find intramembranous ossification? Intramembranous ossification we can find there in the skull. So the bones of the skull is ossified by intramembranous ossification, and the mandible and the clavicle. Here you can see the picture from the skull of a baby or a fetus. Here, these pinkish parts, these are the developing flat bones of the skull. And the endochondral ossification is the characteristic of the growth of the long bones. Here you can see the histological picture of this ossification. The intramembranous or desmal ossification, I told you, we can find it in the bones of the skull. This is a very good picture from the skull of a newborn, where you can see the ossification centers from where the ossification started, and you see that the skulls, the bones, they are not ossified. Only just later, 1.5 years later, because here will remain this connective tissue structure, which is called the fontanelle, here on the top of the skull. And this is really important, because if we have ossification earlier between the bones of the skull, it could be a problem, of course, during the birth. And here you can see some developmental malformations where we have earlier ossification in the transverse suture, so that's why we have an enlarged skull. Or, of course, with special artificial modification, we can change, of course, the shape of the skull during the development. Here is the schematic picture, what I showed you during the desmal intramembranous bone formation. Here is the osteoid, osteoblasts on the surface, osteocytes within the osteoid, and these are the osteoclasts, which will destroy the tissue from the other surface. So that's why when the skull is growing like this, we won't have a very thick skull bone, because the osteoclast will destroy it from the innermost part. In case of endochondral bone formation, it is a special characteristic for the growing of the long bones. We have a hyaline cartilage background tissue, and from the perichondrium will start the ossification of a bony collar, and because of that the cartilage will degenerate and vessels will rupture and will form the primary ossification center, because the protoprogenitor cells will come with these vessels. And here you can see that parallelly with this procedure will develop the secondary ossification centers at the epiphysis of the bone, and here will form the epiphysis, the diaphysis of the cells here. Here is the cartilage on the surface, and here in between the epiphysis and the diaphysis, here you can see the epiphyseal plate. It's one part of the cartilage which will proliferate, and because of this proliferation we have a longitudinal growing in the bone. And if this epiphyseal plate will ossify, earlier, in this case we can talk about the dwarfs. You know, they have a normal shape in face and trunk, but the extremities, they are smaller because we have no longitudinal growing within this bone. This is the histological picture from the different stages. First, we have a normal cartilaginous zone, which will degenerate. And finally, we have a mesenchymal invasion zone, when the vessels will come, the osteoblast will form the osteoid. The osteocytes, they are located within the trabecules, and we can see only just a few little part of the trabecules consists of cartilage, because now the osteoblasts, they produce new osteoid to form the trabecula. And finally, I have a few minutes to talk about the muscles. We can talk about 3 different muscle types: the skeletal muscle, the cardiac muscle, and the smooth muscle. You can see the skeletal muscles will originate from the skeleton. We can activate it with voluntary regulation. They can move the joints and different parts of the body. The unit is a muscle fiber, skeletal muscle fiber, and which is a very special characteristic is the striation. That's why we say that this is a striated muscle. The other muscle is the cardiac muscle. Here we can find it in the heart. We have a very special system which will regulate automatically the movement of the cells, myocardial cells, but we can regulate also with the autonomic nervous system. But this is an automatic system, consists of heart muscle cells, which will also form striation because of the myofibrils, the actin and the myosin filaments. Here we have heart muscle cells. They have X and Y shape, and they will form a syncytial functional syncytium, because between the cells we have very strong connections and they are worked together like a unit. And finally, the smooth muscle cells, we can find them in the wall of the internal organs. In the skin, the musculus arrector pili muscle. The unit is the smooth muscle cell, 3-5 micrometer in diameter. The length is 15 to 200 micrometers, so not very huge. And this is under neurohumoral regulation. If we see the skeletal muscle, I told you that these are the skeletal muscles. We can find it in the tongue, in the esophagus. They show striation, and it is under the control of the somatic nervous system. And I told you that the unit is the skeletal muscle fiber. Here you can see a muscle, and within the unit is a muscle fiber. The membrane is called the sarcolemma. And within the sarcoplasm, we can find a lot of myofibrils. The nuclei is located under the sarcolemma. We have a lot of mitochondria within the cytoplasm. And here you can see the sarcoplasmic reticulum and the sacrotubular system, which are important in the calcium storage and the activation of the muscle fibers. You learn this in physio and biology, the details. This is really important to show you why do we have striation, because the myofibrils within the cytoplasm, they will form a very special shape. The unit of them is the sarcomere, which is located between Z lines. Into the Z lines will attach the actin filaments, and between them we can find the myosin filaments. And because of this very special orientation, we can see the striation within the muscles, where we have a paler band. This is called the I band here. When we have the myosin filaments, these are the A band, and within we have an H paler H band within the A band where we have only just myosin molecules. And of course, these molecules, they are able to move on each other, slide on each other during the contraction, and the sarcomere will be smaller, and this is how we can contract the muscle. Here you can see the steps of the contraction, how the myosin molecules, they attach to the actin molecules and they will move on each other. But it is really important that for the relaxation, we need ATP to dissolve this connection between the molecules, and that's why we have a rigidity after the death, because we have no ATP to solve the connection between the actin and the myosin filaments. Here you can see a picture from the cardiac muscle. The unit of the cardiac muscle is a cardiac muscle cell. So I did not tell you earlier, in a skeletal muscle, the skeletal muscle fiber could be 70 centimeter, depending on the shape of the muscle. But here the unit is a skeletal. Here in the unit is the cardiac muscle cell, which is only just 200 micrometer, which could have X shape and Y shape. That's why they will form a meshwork. But between them we have very strong connection with gap junctions and desmosomes too, and they work together like a functional syncytium. Within the cytoplasm also we can see actin and myosin filaments which will form similar striation, and we can find also a lot of mitochondria, the reticulum, which are important in the movement. And you see that we have very huge nuclei, because these cells, they are not able to proliferate, so they duplicate the DNA content, but they do not divide. That's why we have a very huge nucleus. And finally, here you can see the picture from the smooth muscle cells, what we can find in the wall of the organs, in the vessels, in the wall of the vessels. They are smaller cells, 3-8 micrometer. They have a very special elongated shape, and under neurohumoral control they are. We have also actin and myosin filament within the cytoplasm, but they do not form striation. They are able of very huge contraction, which can keep it for a long time. And that is what you can see in this picture. We have a very special cells too, which are called the myoepithelial cells, which could have contract, but they are originated from the epithelium. We have to mention in this case, when we are talking about these cells able to contract, these myoepithelial cells, they are located around the glands. They can help in the secretion of the glands, but they have epithelial origin. So that is what I could tell you within this 45 minutes about the connective tissue and the muscle tissue. I hope that you could learn the main basic details. I hope you can understand the main characteristics of the tissue. I wish you a very good study for that.